Okay.
Or work something very simple, and I was worried, so I was like, well, I just like even like X amount of times, like, I didn't notice where to go.
If one by one. Like everybody else. Yeah.
No. All right, 6 o'clock, let's go ahead and get started.
But still have what it's working.
We're getting started. Okay, so all right, so you might have noticed, or hopefully it's visible, I reopened all the owls.
Yay, you're awesome.
If you submitted it though, and you got 100%, I didn't reopen those ones, and I'm trying to get it so that way you can use them as practice, but like not change.
Count toward, yeah.
Yeah, and I'm struggling to figure that out, but it's coming, I'll figure it out.
So one of mine came back, and I thought I had 100 on it, but it came back.
Yeah, I know exactly which one you're talking about. A lot of people, it was the third one, right? A lot of people, it was out of 29, for some reason, I don't know, I gotta figure that out too. And it was 27.19 out of 29, so I reopened those ones, but I find it weird because a lot of people got 27.19 of that. I don't know what was going on there. Like what? This not even making sense.
Because I looked at it and all of them were answered.
Oh, really?
I submitted it back, yeah.
Oh, that's fine. I'll figure it out. I cannot express to you how difficult it is to use Owl on my end. It's like, like, oh my gosh. Okay, so there's 2 different discussion groups. There's 2 different Owl like groups, so I have to go in and change the settings. Well, I thought I had changed them both for both groups, and then on Sunday I was like, can you guys all see the owls at our office hours? The people from one group said yeah, the people from the other group said no. I was like, okay, I'll go in there and fix it. I go in and the settings are the exact same, like I have the exact same boxes checked on both, and I was like, why? Like what am I doing wrong? Okay, but anyways, so I want to open them all back up, and you guys can submit them for no late penalty. So I want you guys to be able to go back, even if you had like a 93 before, and get that last question, because I want you guys to, this is only really practice you get before the exam. I want you to do that practice. I don't want the whole time thing to, discourage you from doing the practice. So I have a practice exam written. I plan on posting it tomorrow or Thursday, whenever I get my whole, I'm doing one more announcement this week. I'm gonna post the practice exam whenever I finish whatever lecture we don't get to. Hopefully we get through it all. And then I also, Destiny requested that I make kind of like a summary of all this naming stuff and kind of put it in one. I tried to make it short, so that way it's like a cheat sheet almost, but then I thought to myself, like, it's kind of a lengthy document, because I feel like there's all these like weird exceptions that I wanted to put in there. So I'll have that posted again Wednesday or Thursday. And then I plan on, so you guys can, the practice exam I post, you can practice it anytime, email on questions at any time. But the office hours before the final, so not this Sunday, but next Sunday's office hours, I was gonna make it a 2-hour from 6 to 8, and specifically go over practice exam stuff, kind of as an exam review. You can ask questions and stuff like that. We have our last in-person lab tomorrow, last lab grade period. There's no more labsters or anything after this. And I just want to remind you all that any of the in-person labs, I'm offering 50% credit back to any point if you miss. Just the caveat is you got to explain to me why that answer was wrong or what the right answer is. Like, I want you to learn from it. But I do want to offer that.
So I can turn in my lab from last week and just.
Okay. Did you look at the score? You're like,
Uh,
never mind.
Okay.
Alright, so this week we're covering 4 chapters. Crazy, but we're doing biomolecules. Which hopefully is. Do you guys have to take biology for this?
Michael. Before you already put on it.
Okay, so hopefully this sounds a little familiar, unless they didn't cover it in this biology course, but typically these are covered in biology courses. So these are all categorized as biomolecules or biological molecules, right? So we're starting to move into biochemistry. Which is exciting, that's what my degree is in, I'm excited. So what is a biomolecule? These are molecules that are essential. I'm gonna spell essential, there we go. To one or more biological processes. Specifically metabolism. So all those functional groups that we've been talking about the last 2 weeks. Those are really strong characteristics that make up these different biomolecules. So we have lipids, we'll talk about them, nucleic acids, carbohydrates, proteins.
Are you saving the notes this time?
I will, I'm pretty sure I know how to do that, yes. All right, so another characteristic of biomolecules is that they are. I probably have to turn it off. Thank you.
All right, I'll just get the email.
Okay, I'm sorry, it's gonna take a minute.
Look at the schedule. What did you say? What you taking that? I just have, because I think that.
Yeah.
Season.
Tuesday and Wednesday. Oh, I don't have to say Friday. On Friday, yeah.
What three you like? Listen to that.
Baby genius over here. I mean, I know you talking to her. About maybe. We find out what. To get the sun away.
No, look, this is happening.
She had to do that.
Major technical difficulties.
You want to see one of the questions?
I want to discussion.
Like, you gotta do- You see them coming up, immigration and all of that, you know. I don't even have my bag of cords, that's all right. I wouldn't-
I already got that. See, I say that. I say that. I'm gonna do better. I'm trying to go to you.
Did you check the input? You like it good, but
Especially clean. I did look at the angle. I say half the glass, but it's 2 glasses.
Yeah.
Turn it off. It's not like-
No sleep.
Yeah. The,
It's great. That's it.
So.
Go back to. Let's, I gotta go on board. Let's see if I can-
like, the,
So you got it?
one that goes,
So you got it going.
Not really.
Control V. Control V is not. I want to say this, that, you know, that's why I got hired.
No, I'm scared.
Yeah.
It's not bad.
right, that is like the bottom, yeah.
Then you'll get the signal.
I don't know what's like. Do you guys have Wi-Fi? Is it a Wi-Fi?
I think that's Bluetooth. Well, you gotta be on the same Wi-Fi.
That's how much, this is what I'm saying.
Wi-Fi, do you use?
I use the staff.
Use the staff, okay, that's why I can't catch. Right, yeah, on staff.
Not in May, but what you have, the one in Jordan.
I'm gonna look back on. I think it's probably-
Yeah, so we turn that off, back on, we'll turn this off, back on. It just stopped mirror playing and it now it won't go back. And then I tried to plug it into the HDMI. still it didn't. I have a video. Yeah.
So basically.
Yeah, so I have this down, but I'm not sure if I'm down here. I've never tried.
Oh, this black piece? You
Yeah.
gonna use that?
Oh, you have. Yeah. The best part is the second, the first thing I bought also.
I'm charging you. iPad change. Little bitty one. The normal phone charger.
Alright. I do play, I either plug in my computer and project the slides, but then I can't write on it. Or we can go to another room. What do you think?
Yeah.
Let me see that cord again.
Well, he was gonna unlock the door. He wanted to try to play around. Yeah. Probably. Let me figure out if it works. Oh. Gonna figure out first.
Somehow in my ear.
One piece with the white, please. It's her piece, it's not that. It's her piece.
Move over there.
So it ain't this, it's
Okay.
this.
Oh.
Yeah, this piece, like, so when I plug her power charger
Yeah.
in, I got signal. But when I
Right.
plug this in, into this, and then plug the power into there, it didn't power up.
Oh.
So it's this piece, not this piece.
Huh.
Yeah.
Well, thank you.
Have you used the white piece before?
Yeah, I have.
So I got one similar, I got a
Yeah.
small one, a 2 one, just the
Yeah.
power and the HDMI.
Uh-huh.
It's fun. It's fun. All right, we got next door.
Yeah.
Oh, okay.
Okay. Sorry about that.
What's the day? He was already gone for the day. He should be leaving. Know that it's time to go.
I don't, like, it's lunch for 5 weeks and then there's no.
It happens all the time.
Really?
Yeah.
It really freaks me out.
It's not, I mean, it's happened on.
It's like my plan, he wasn't on the work and.
What are they doing? They just chatting with him? They're coming out. It's funny, I got.
Is that everybody coming? Good point. So characteristic, let's get going since we lost some time there. A characteristic. All right, we're gonna get rolling since we lost some time. So a common characteristic of biomolecules is that they're tiny pieces built together like a string. So we call
It
this,
looks like a screen, that's a screen.
yep, so they're tiny pieces string together. So we call these tiny pieces monomers. They're repeatable units. And then one big string is a polymer. So each of these biomolecules have their own monomer. And then that unit is unique to a nucleic acid, or this monomer is unique to a carbohydrate. This monomer is unique to a lipid and unique to a protein. And then they are then repeated, create the entire molecule itself. So, what was I gonna write here? Okay,
Chapter is this,
this is just my introduction.
editing, needs to.
Tell me now that my pen's not working. Okay. So biomolecules are polymers. Made up of monomers. Okay. These are repeating pieces here of this whole thing. All right, so those two slides were just kind of these characteristics fit all of the next chapters we'll be talking about. So chapter 17 is carbohydrates. So what the heck is a carbohydrate? So a carbohydrate is a carbon-containing molecule that's composed of carbon, hydrogen, oxygen, typically in the ratio of 1 carbon to 2 hydrogens to 1 oxygen. And a characteristic of this is that they contain the functional group either a ketone or an aldehyde. So they either contain some sort of C double bonded to an O at some point in this chain. So it's an aldehyde if it's on a terminal carbon, which this one happens to be. If it was somewhere else, say it was in this middle carbon, then it would be a ketone. So carbohydrates typically end in ose. So carbohydrates, glucose, sucrose, fructose, lactose. They typically end in ose. These are sugars, fatty acids, and starches. Right, so bread, potatoes, yeah, there's carbohydrates and fruits, because essentially carbohydrates are also sugars. So we said that, there's the whole characteristic of all these biomolecules is that they're small pieces chained together. So one monomer of a carbohydrate would look something like this. And then if we were to string those all together, we would have a carbohydrate. So we've given special names to monomers that are specifically carbohydrates. And that is a monosaccharide. If we had 2 of these repeating units, 2 carbohydrate monomers, that would be a disaccharide. And then, just ignore this one, I don't like that they put that there, but basically a polysaccharide would then be the equivalent of a polymer, but just specific for a carbohydrate. Right, so a bunch of those units repeated. So, yeah. Okay. So a characteristic of carbohydrates is sometimes we'll see them in the cyclic form, and specifically, they'll have this like kind of ring put on its side. They're trying to show us that this is 3D and that some of these OH groups come off the top and some of them come off the bottom. So if you see something like that, that's still a carbohydrate. It's basically just a broken down form of this chained together. Okay. So carbohydrates typically form rings. And that's what we're looking at here. So if we were to bond multiple of these rings together, we have a carbohydrate biomolecule. Specifically, we can come up with a specific name for the bond that joins two monomers of a carbohydrate, right? So this is a glycidic bond or a glycidic linkage or a glycidic link, all synonymous. So this is a bond between monomers of carbohydrates. I'm gonna abbreviate carbs or carbohydrates as carbs. I think I did that on the last slide without explicitly saying it. So these glycidic linkages or glycidic bonds have specific names based off of if it's an OH group going up or coming down. And so here we have two sugars coming together and forming sucrose. And this bond here, glycidic linkage, we can give it a specific nomenclature, if you will. So bonds that go down we give the nomenclature alpha, and bonds that go up have the nomenclature beta. So if I were to name this glycidic linkage here, I would call this molecule 1 and this molecule 2. They both happen to be going down. So alpha 1, that OH bond is going down. Beta also 1. Why the heck you need to know this? I don't know. Oh, I'm sorry. Okay. It also going down, so also alpha. Alpha 2. I don't, why the heck did I? So, yeah, I'll show you an example. Okay, so if we went to the back to the first slide, let's say this was molecule 1 and this was molecule 2. This is coming down, but this one is coming up. Here they depicted it side by side and they had to throw them kind of in a V to show that they're both going down.
Oh,
Yeah.
that with the oxygen.
Yep, yep.
They're showing that it's going down.
Yeah.
Oxygen's at the top, so I need to get one down. So that
The way they're connected. it's-
Yeah, like the bonds are coming down.
that's showing me. Okay, so
Yes.
it was pointing like upside down.
Like it was the opposite
Yep.
direction.
It'd be going up.
Yep,
That if they're side by side.
yes, and if they're up and down,
I
so,
don't know.
so if you had one going up and one down, though, you can't really depict it side by side, and that's when you have the up and down, like,
Right.
up and down molecules. Did you have a question?
Yeah, I'm sorry. Which oxygen are we looking at to
Yes.
determine, like,
One in the circle.
Here.
one in the middle, and that's always?
Yeah.
Yes,
Okay, that's what.
If they side by side, that's how.
yeah, yes, here, I believe I have another one right here. Oh, I forgot I threw this one in here. Okay,
Ain't going nowhere. What they doing?
It's on the way down.
so they're trying to show you that this one's going up and that this one's coming down.
Okay, I see it. Why explain it like that?
Oh,
Yeah.
okay.
So if this was the molecule one, and it's going up, this would be beta one. And if this is the molecule coming down, second molecule would be alpha two. There's only one practice, or one question like this on the exam. And I don't like, I hate that they asked this, like, what does this do for you? I don't know.
Is the accuracy going up or down?
Yeah, you have to, you'll have an A, B, C, D, and it'll be like alpha one, alpha two, beta one, beta two, or is it an alpha one and a beta two, or is it a beta one and a beta? or is
That's
it
what I use.
Yeah,
Wow. Can you, put on your screen, one, which oxygen you looking at? I'm just.
yes, let me highlight it actually. I'm looking at it.
That's for, one.
That's one.
Mhm.
So we're doing one.
So, no, we're looking at the oxygen that binds both of these rings together. Does that make sense? Yes.
So how do we know if the one that goes up is beta or not?
Where's-
So up is always beta, down is always alpha.
Yes, it was like this the other way.
Then the first one be going down. the second one be going up.
Right.
Well, how-
Where's the second one at?
So,
the oxygen that's in the middle is
Yes.
determined for both of them. That's what
yes,
you're saying, right?
so,
Oh, cause I'm thinking it's two oxygen somewhere.
That's why I had.
Where's the one at?
yeah.
Okay.
So-
Sorry, yeah, so one oxygen that combines those two bonds, or the two molecules, yeah.
So
Where did you get that data from? I know you heard.
So, this is the first molecule. And because this bond,
Oh, because it's going up, it's
Yep.
beta, and if it go down, it's alpha.
Yep.
Got it.
Yep.
Okay.
Oh, okay.
Okay.
Two of them.
right,
Only in the middle. Is one up and one down. Oh, okay.
so if we saw this on exam, right, and these are all our answer choices, we would choose beta one alpha two, and my Greek there is a little wonky, but.
Can I write that on the side just so I have it?
This is not the exam for your test though.
Okay, I just wanna be a.
Yeah, yeah.
What's going for the test?
I'll help you, I'll give you, I have a list of questions that I'm gonna give you and
Maybe
last,
this one, you know?
cause I think once you get it, you'll get it.
Yeah, it's easy.
Yeah. No, my list last week was, or midterm was like, I gave you one and I just had to correct two is form, right? My list right now is 4 and then
Only
correct 2.
Come on.
You said it's 44?
I'm reading over the exam, right, and I have 4 questions that I wanna give you, because I think the answer's either wrong, or the question is just, it's just the most loaded question, because the way it's worded is just horrible, right?
Right.
That's very generous.
Yes, it is. Yes, it is. You're off. On
Yeah.
time.
Alright,
You're awesome.
so, some common physical properties of carbohydrates, right? They're typically solids at room temperature, right? Typically, right, but we have some sugars, sometimes you get like the fructose syrup or something like that, but typically, right, we think of potatoes, carb in bread and stuff like that. They're also extremely soluble in water because they have that OH group, right, so we. This is an alcohol group. Right, we talked about that in lab 5, right? That was very soluble.
So are you saying OH is, a carbohydrate?
No, OH is a functional group that is common to carbohydrates.
A floating.
Functional group, right? Right, so. We said that they're either aldehyde, they either have aldehydes or ketones. Right, which means at some point they had a C double bond O. And they also typically have OH groups or alcohols. Right, and I'll come back to this slide, but here we had OH group. If I went back and showed you, it's kind of hard to tell when they're in the ring structure, but here was a C double bonded O, OH group, right? There. They have character, they have those functional groups, that's a common characteristic. So some important carbohydrates. We have ribose and deoxyribose. So here's an example of ribose and deoxyribose. Don't memorize it, just know that these are sugars. And that these are important because these are the backbone sugar in our DNA and RNA. So DNA, if you ever wondered what it stands for, deoxyribose nucleic acid. This is where the deoxy, the D in DNA comes from. It's because that's the name of the sugar. Where RNA, which we'll learn about next week, ribose nucleic acid, that's the backbone sugar there. Okay. So we'll learn a little bit more about those next week, but I want to introduce them. Another very common important carbohydrate, glucose, it also is known as dextrose. This is the only type of sugar that our bodies really carry, right? So this is when we take our blood sugar, we're measuring the level of glucose. So everything in our body must be converted to glucose before it can be used for like metabolic processes, right? Another common carbohydrate, lactose, right? So people have lactose intolerance, all right? So this is known as the milk sugar. This is what it looks like. It's a two ring structure here. This would be a beta 1, alpha 2, right? You don't have to remember. I just was.
I thought I didn't have to put that.
All right. Sucrose. Now, when we go get like sugar to bake, that's the kind of sugar, that's gonna be like our household sugar, sucrose. It looks like this. It's a 2 carbon, I keep saying carbon, 2 ring structure. Alpha 1, beta 2, right? This is important because this sugar can't be reduced. This glycidic linkage will not open. We can't break that bond. It's important that in sucrose, we can't reduce that bond. So this prevents them from ever being monomers. They're always those 2 rings together. Lastly, we have cellulose and glycogen, right? So these are 2 storage sugars. These are storage carbs, right? So I said that, you know, our body has glucose, but at some point we might have too much glucose, but we want to store it. We change that glucose to glycogen. So for animals, our storage carb, the carbohydrate that we keep in storage basically, is glycogen. Where in plants, it's called the same, not the same, but the similar sugar is cellulose. Specifically, just a fun characteristic of cellulose is it makes up the cell wall in plant cells. So when we eat lots of fruits and vegetables, we're eating cellulose. So these are similar because this is the way that we keep, you know, the stored form. But some characteristics here is that glycogen is very branched and cellulose is very orderly and rigid, right? Kind of reminds me of celery for some reason. I don't know, it's just like nice and rigid. Carbohydrate, oh, I'm sorry, glucose specifically has an alpha 1,4 glycidic bond. And glycogen has a beta 1,4 and a beta 1,6. So I don't like the way these are written, really. This is really alpha 1, alpha 4, beta 4, or beta 1, beta 4. And so
Is that a plus in between or and?
I'm gonna read it. Okay, so if there's anything to remember about the difference between cellulose and glycogen, their bonds are different. Izzy got one of them. So there's two metabolic processes that, again, we'll talk a little bit more next week, but I just want to introduce the idea that we can change glucose to glycogen, right? So this is floating around in our blood, and this we store in our liver. The process of going to storing it, basically, from going from glucose to glycogen is glycogenesis. The process of reverse, going from glycogen to glucose, is yeah, I'm not gonna say.
Is that the breakdown of sugar?
Yeah, so this is the, I like what you said, but I want you to be, this is the breakdown of glycogen. Glycogen is a sugar, but yes. So diabetics have a hard time doing these two processes, right? They have a hard time regulating the blood sugar, whereas people who aren't diabetic, this process is functioning normally. So let's say I eat a cupcake. My body's like, you don't need all this sugar. We're gonna do glycogenesis, store some in your liver. 6 hours later, I go for a run. It's like, oh my gosh, we're expending a lot of energy. We need that sugar now. Pull it from the liver. And glycogenesis. Yeah, okay. All right, so that's our summary of carbohydrates. Moving on to lipids. So what is a lipid? These are organic compounds, they're biomolecules. And a strong characteristic about them is that they're insoluble in water. They're typically composed of a head and a long hydrocarbon tail. The one exception to this is steroid and waxes. So if we look at triglycerides here, we have this head group, and then we have these long tails that are composed of hydrocarbons. And if we remember from the saponification lab, right, we were taking a triglyceride, which had that long fatty acid tail. We were trying to make soap, kind of a flop. I don't think we got the reaction hot enough for the reaction to happen and then cool it in soap. Yeah. Well, so yeah, after you guys left, Alyssa did put it in the refrigerator and brought it back out like 10 minutes later, and it had solidified, but I think it was solidifying back into the coconut oil because it was still like kind of like two different. Yeah, it didn't feel like soap. So I'm thinking, somebody had pointed out that the hot plates weren't really getting that hot. So I'm thinking,
They only give like 75 degrees.
yeah, it never got to the point in which the reaction happened and then solidified into soap. So they had problems with this lab before, I heard. So
What are we doing tomorrow?
tomorrow we're doing, it's kind of cool. We're gonna do these like little tests to test for the presence of biomolecules, so there's these different tests, and we're not gonna learn about the test in lecture, I have a little like pre-lecture tomorrow. But you basically put a drop of, let's say, I'm gonna bring some yogurt, so put a drop of yogurt, and then these different reagents turn a different color based on if there is a protein present or if there's a carb present. So, yeah, I think it'll be cool.
You don't decide right now.
No, I want to say a few more things. So, yeah, so composed of a head group here and the hydrocarbons, right? So those hydrocarbons, right, something about, if you remember, those are the long chains of alkanes that are just strictly hydrogen carbon. They're very nonpolar and makes them very insoluble in water, right? So it kind of brings it all back to solubility here. So the three kind of categories here are phospholipids, triglycerides, which are also called fatty acids, and then cholesterol, which can even be broken down further into steroids and waxes. Okay, now I'm done. So we talked about saponification last week, which was changing these triglycerides or these fatty acids into soap, right? Well, there's some lipids that can be saponified and some that can't, right? So this is just a chart of what can and can't be saponified. Right. And so last week we were using triglycerides, coconut oil. If we wanted to, we could do the same thing with waxes and these other two that we haven't really talked about. But what we can't saponify is steroids and prostaglandins. That's all you need to do. Can't saponify those two.
Wait, 1 second.
So phospholipids, hopefully we've heard this term before. Yep, from biology. Right, so these are what mainly make up our cellular membranes. Right, so phospholipids, you know, previously I just kind of introduced the idea that they had this head and these tails, but more specifically, phospholipids have a phosphate group. Right, this is a phosphate group. It's, if you remember, this is one of our, E3O minus, this is from our, what was that list called? The naming and the, I don't know why I'm blanking on this word right now. Polyatomic ions, right? So they have a phosphate group. They also have this glycerol. Together, these make a hydrophilic head. So what the heck does that mean? Hydrophilic means it likes water. And usually things that like water are polar, and this is. So this is a polar head group that likes water. So then we have these tails that are alkanes or alkenes. They're all made up of carbons and hydrogens. They're very nonpolar, meaning they dislike water. Right? Hydrophobic means you don't like water. So if these make up our cellular membranes, kind of doesn't make a lot of sense, right? Our bodies are made of a lot of water. So these phospholipids arrange themselves. I'm gonna say tail to tail or butt to butt. So that way these polar head groups that are hydrophilic are facing the cellular contents, which is mostly water. I have a lot of writing on this slide, I'm sorry.
No.
But basically saying, the head is, pointing towards the water because the tail is like water.
Yep. So together, we call this a bilayer.
So all heads like water, tails don't like water.
Mhm. I still have more. Alright. I was gonna just write more. I was just kind of letting it soak in.
Oh, okay.
So, a thing to point out here in these fatty acid tails. We can have saturated fatty acid tail or an unsaturated fatty acid tail. And so saturated means that it utilizes all 4 carbon bonds. So it utilizes all, if remember, carbon was special to organic because it could form 4 bonds. So a saturated fatty acid is utilizing all of these bonds. So if we were to draw out the implied hydrogens, right, there'd be 2 here, 2 here, and so on. Where an unsaturated is not utilizing all of them. So basically, there's the presence of a double bond. Right, so saturated has maximum amount of hydrogens it can have, basically. Unsaturated has a double bond, meaning that it doesn't have the maximum amount of hydrogens because instead, the 4 carbon rule tells us that there would only be 1 hydrogen here and here. And now I'm done writing on this slide. Saturated fatty acids are all single bonds. They utilize all the possible hydrogens that they can have. So when 1 carbon is singly bonded to the carbons nearing it, it has the maximum amount of hydrogens it can have, which in this case is 2.
And that's saturated.
Saturated. If I were to create a double bond, I then have to take away one of these hydrogens because carbon has to maintain that 4 bonds. So it's now unsaturated, doesn't have the max amount of hydrogens it can have. So I think how I worded this was a little misleading. I'm gonna say utilizes all 4. No, how do I say this? Has maximum amount of hydrogen they can have. Does that make more sense? So why that's important though, so this picture that I showed you, all the tails look like they're probably saturated. There's no kinks in them. They're aligning nicely. But it's possible that some phospholipids are saturated and end up having a kink. And that causes the membrane to end up opening up in some places and kind of having gaps. So that way ions could flow through, proteins could flow through. So that brings us to the fluid mosaic model, which basically this is the model in which our biological membrane follows. So these hydrophobic tails are kind of wiggly and, you know, because some of them have kinks, some of them don't. And when you arrange them together, you start to get more 3D looking, and you get pores basically. Wiggly, it's mosaic. So our cell membrane is mainly composed of phospholipids, but not exclusively. We have cholesterol hanging out, we have proteins, we have carbohydrates, but as you can see, it's mostly these heads and tails. It's mostly phospholipids, but not exclusively. I don't know if I spelled exclusively. I know I didn't spell it right. But yeah, just now.
One of those words.
So this makes it where large molecules they can't pass through this. Through membrane. Yes, this is a characteristic. Yeah. More so the membrane itself. So this idea, that's, we're gonna ask you what that says. So let me just start. So this idea that large molecules, should have done that. Large molecules can't pass, but small molecules can, like ions, they could flow through this channel, stuff like that, water, makes this a permeable, right? So some things can, it's permeable membrane, some things can pass, some can't. Right. So characteristics of biological membranes, to summarize. They're mainly made up of phospholipids. Phospholipids typically form this bilayer with the hydrophilic heads to the outside and the hydrophobic tails to the inside. They're permeable, so that way large molecules can't really pass through, but small molecules can. And it's not necessarily that it's exclusively phospholipids, right? Our membrane is fluid and mosaic. We have crinkly tails, we sometimes have proteins and carbohydrates, but it is mainly phospholipid. So moving on to our next type of lipid, triglycerides and fatty acids. Again, they're made up of some kind of head and then a hydrocarbon tail. So here's our head. And it's depicted down here. And then our tail, which is depicted down here. Right, so that tail is mostly hydrocarbons, right? So it's very nonpolar, doesn't like water, where that head is hydrophilic and likes water. So the difference here is that the phospholipid, specifically its head was a phosphate group in a glycerol molecule. Here, triglycerides are a little less specific in that they're just some kind of polar head and a tail. So what we were trying to do with the saponification reaction was to drop the hydrogen here, create some kind of carboxylic salt, basically. So we would get some kind of ion here. And basically, this is what soap does. Is it arranges itself with all the tails facing each other, and then all these polar heads facing each other, and it engulfs whatever you're trying to clean, basically. So these are called micelles, so like soap micelles. So you're trying to clean your stuff, basically traps all the it basically traps all the debris. But if you've ever had something greasy, right, it's not very soluble in water, so sometimes it takes more soap to trap it all up in here. I just think soap micelles are cool. They're not necessarily strong, but they're just cool. So fatty acids, if we were to depict them more in this, I
MICEL, milestone.
think that's how you spell it, yeah. MISCL.
Flip back, 2 seconds. Go back. Yes.
There's a Y on the commercial.
I think so. I feel like I don't know.
Soap micelles are, I just think they're cool. I think they're applicable in real life. Yeah, it's just an example. They're just cool.
We should make something instead of doing it. You gotta do it right. At least one burner.
Well, yeah, it's like, we need that. They're just hot plates from Amazon, that's what I heard. Like,
I think it's because we had 12 on the thing.
and yeah, we need to spread them around too.
Yeah.
So if we're to depict fatty acids, in this like modular form here, where we have that polar head group here, these are the oxygens and the hydrocarbon chains. There's all different kinds of fatty acids. There's, I don't know if I'm pronouncing this right, stearic acid, oleic acid, linoleic acid. And basically, the more double bonds you add, the more you decrease the melting point or the boiling point. And this was something we learned in module 4, where alkanes and alkenes are both very low boiling points, but alkenes were slightly less, had a slightly higher boiling point than alkynes. This is an owl. I think this has something to do with owl. So as you increase your double bonds, you lower the melting point or the boiling point. There,
So increasing double bonds lowers the melting point.
Yep.
Well, so next point is the boiling, BP same thing.
Yeah. This is just to get you through an owl question, basically. This is not necessarily on the exam. Yeah.
Mm-hmm.
Yeah, so I guess I should clarify. boiling point and melting point, I do believe is on the exam. It's one of them that I'm giving you the answer because, again, the no correct answer is listed. I think whoever, why is nobody checking? I don't know. Why don't they like give them to a friend and say, look over this for me? I don't know.
Or they wanna figure it out.
Something, I don't know. But this is in the owl. So I'm gonna skip these next two slides because they are already slides I showed you in the free lab last week. So another type of lipid is sphingomyelin, right? This is just another type of lipid. So a characteristic of this is that we have this sphingosine head. Don't really know what it is, but just. And this is attached to a fatty acid tail and a carbohydrate. It's bonded here by an amide bond. That means it has C double bonded to an O, bonded to an NH. So sphingomyelin has a sphingosine head attached to a fatty acid and a carbohydrate by an amide bond. Now, phospho.
Is there NH2?
Yeah, NH2. So we're gonna compare this. Sorry. We're gonna compare this to phosphoglyceride, which again is just another type of lipid. This time we have fatty acid tails. We have a glycerol head. But this time the bond that joins them is called a phosphodiester bond. So, how do sphingomyelin and phosphoglycerides differ? Sphingomyelin has amide bonds. While phosphoglycerides has phosphodiester bonds. Phosphoglyceride. All right, so moving into our next biomolecule, proteins. what the heck is a protein? So, this is a polymer chain, right? Going back to the first line, we said all of these biomolecules are these chains, but this time they're composed of amino acids. So amino acid is that repeating unit. Or more general, it's the monomer. So proteins are super abundant in our body, right? They're the basis for a lot of things, such as hair, skin, enzymes, antibodies. The list goes on and on and on. This picture here, this is a protein, right? We'll come back to this picture. Right, so protein is made up of amino acids, right? It's the basis for a lot of things in our body. So, before I switch to the next slide, right? So protein is all these repeating units of amino acids. But specifically, an amino acid is made up of these components. An amine group, here sometimes called an amino group. A carboxylate group, which we called a carboxylic acid, same thing. And then some kind of side chain. This changes. It's a variable. So the R group or the side chain is how we make each amino acid unique. But for the most part, amino acids the same repeating unit, just with this one variable. Does that make sense? Okay. So what's the side chain, right? What the heck's the side chain? You're gonna yell at me if I go to the next slide. All right. So side chain are these, right? So we have our carboxylic acid group, our amine group, and then this is R. You don't have to memorize these, but basically, these are your R groups, right? And you can see how they vary based off of what amino acid we're talking about. So hopefully maybe some of these sound familiar. Tryptophan, that's supposed to be the amino acid that makes you really sleepy. But we have things like proline. I'm trying to think of maybe other ones you've heard of. Histidine, maybe? Histidine, but probably some derivative of histidine, right? So these are the amino acids. They all have the same backbone, right, which we just described was that carboxylic acid group. And then the amine group. But then we added this side chain, right? And that's what made it each a unique. So we have, some amino acid here, right? We have amine group, carboxylic group, whatever this is, some other amino acid here. But we talked about the fact that these all come together to form a protein, right? And so they come together and form a bond. Does this look familiar to you when I write it maybe this way? And he's nodding here. What does it look like? An amide group? Right? Yeah. Yeah. Right. So this bond is called an amide bond. also sometimes called a peptide bond, interchangeable. When these two amino acids come together to create an amide bond, they end up releasing water. This happens over and over and over until we have a polymer or a protein.
So.
Yeah.
How can we have a polymer if we have two different types of amino acids linking together?
Yes, so I was a little misleading. So each monomer is a repeating unit. And it's unique to, let's say, a protein. So a protein has the same repeating unit with variation, basically. But a monomer of a protein, an amino acid, is not the same makeup as a monomer of a carbohydrate. Does that make sense? No, your eyes.
Yeah, I mean, it does, but I mean. It does, so, yeah, so you can have different amino acids linked together to form a protein.
Yep.
But we still gonna call them monomers.
Yep, but if
Polymers,
polymers, yep.
okay.
But if I were to just like throw in, all right, so we said a characteristic of amino acids, right? It's always a carboxylic acid and then an amine group and then some kind of side chain. What if I tried to like connect this to some structure that was only carbons, hydrogens, and oxygens in the ratio of 1 to 2 to 1?
So you try to connect that to a structure that was only carbons and hydrogens.
And oxygens.
And oxygens?
Right, that wouldn't work, right? Those are two not the same monomer.
Okay.
That's what I was getting at when I said monomers are the same. They're the same for each biomolecule, but each monomer itself within the biomolecule has some variation. So carbohydrate, it's in that ratio of 1 to 2 to 1. So glucose is C6H12O6. I know that one off the top of my head, but I don't know other ones off the top of my head. But let's just, for instance, say fructose is C3H6O3, right? So the monomer, there's still monomers of carbohydrates. They still have that characteristic that they're composed of carbon, hydrogen, and oxygen in the ratio of 1 to 2 to 1. But there's variation between.
Right.
Same thing with amino acids, right? They're always going to have that amine group, the carboxylic acid group. Variation is what's the side chain, right?
So will it at least form a pattern? Or no, it could just be.
Yeah, the backbone will always look the same. So we have essential amino acids, right? So sometimes these are like things that you get in your vitamins and stuff, right? And I put a list here in the main food sources that they come from. You don't need to memorize this. What you need to know though is that essential amino acids, they're essential to our body, but our body can't synthesize them ourselves. We have to get them from food source, right? So like, seems like eggs have almost all of them, but so eat eggs, I guess. So they're essential because they can't be synthesized for our body. We have to eat them. They have to be in our diet. So going back to this chart, but now we're looking at it in a different way. So at first I was just showing you this chart to show that they all have these same backbones, but then they have a different R group. But now I want to show you that each amino acid has a unique name. So it has a unique name. It also has a unique 3-letter code, which is kind of an abbreviation. And even further, it has a unique one-letter code. Right, so what's a relatively, okay, so like a proline, right? We have our amine group, we have the carboxylic acid group, here's the R group. It's called proline. If I wanted to shorten it, it'd be pro, and then if I just had said P, we know that stands for protein, or proline, not protein, sorry. So you don't need to necessarily memorize its table or anything, but you do need to know how to use it. So you'll get out questions like this, where it's asking you, what amino acid is this? So you're probably looking at this like, how the heck does that look anything like the table? It doesn't, because it's oriented different. I'll give you that. Let's figure out what our group is. So let's rule out the other stuff. Basically, this is our carboxylic acid group. I'm actually gonna do this in red. Right, so this is our carboxylic acid group.
So before you go, is it just like the regular, structure, the bins are the
Yep,
C's?
yep,
Okay.
yep. We have our amine group here. And I'm highlighting this because this is the carbon that connects them. So that leaves us with this as our side chain. So you have questions on OWL that ask you to identify the amino acid. So you have to be able to first identify what even part of this molecule is the side chain. And so here we have it's a carbon to an OH. So if I were to go back to this, I'll give you a hint.
Purine?
Yep. Most of you guys did that without even seeing that highlight. Yep, so it's a serine, right? So name this. This is the amino acid serine. It has a 3-letter code of SEER, which I think that's what Al likes you to type in. As a 1-letter code of just S. It's asking you just to select the carbon.
It says select the alpha carbon in this amino acid. Yeah, so everybody's might be different.
Okay, so you said 3-letter code.
I have to brush up on what now. Not gonna lie. I think it's this carbon. I think. This is carbon rice amino acid, so how this is I'm just gonna ask him. Okay.
Let's see what else.
All right, let's do another one. So here's my carboxylic acid group, right? That's not part of my side. It's part of the protein or the amino acid, but it's not part of my side thing. Here's my amine group. Oh, why did I just do that? And then here is my side chain.
That's the.
Yeah? Oh my gosh, Kyle, you're just way ahead of me. Yeah, so this is histidine, right? And Kyle, how'd you know that? You went to the chart, right? All right.
Hold on.
Oh, yeah, I see it.
First one, like, amine group.
The red is the amine group here. Now, histidine has multiple amine groups. But what I find a characteristic of histidine, let me show the laser here, is this ring with the nitrogens in it.
So you said the red is gonna be.
Yeah, so what well, let me. get more specific. This part of the red is the amine group. This part of the red is the carboxylic acid group. I put them in red because they aren't the identifiers here. But when you want to identify an amino acid, all amino acids are gonna have an amine group and a carboxylic acid group. Right? That's the backbone of amino acid. The identifier here was what's in green, and that's your R group, right? And you gotta basically just go to that table.
Yeah.
All right.
It's the main one.
Sure. It's an important part of it, right? It's what makes it unique. All right, I know we normally take a break. Do you care if I try to push through since we had that technical?
Yes,
Yes,
please.
yeah, push through. I don't wanna- Well, yeah, we'll take it.
Wait.
So maybe you can leave early.
Maybe, I don't know. We're getting.
Yeah.
All right. This is like my favorite part about proteins. So proteins end up forming this unique globular structure, right? So I showed this picture to you earlier, right? So if I were to yank a protein apart, sure, it'd be these beads on a string, one by one by one. But they have this really unique folding that they end up doing, right? So there's 4 phases to this fold. There's your primary structure, which is sometimes designated by like a 1-degree symbol. You have your secondary structure, again, like 2-degree symbol, sometimes you'll see it like that. Tertiary and quaternary. So proteins have the ability to fold and create unique structures. Specifically, there's 4 layers of this folding.
Is that all that was on it? You got 4 layers of foam?
Yep. So now we're gonna talk about how each layer basically works. So primary is basically just the primary structure is how those amino acids are lined on that beads on a string, basically just one by one, right? So, this is my backbone, and we have my different R groups, right? So these could be anything. So primary structure is just how they line up one by one. So maybe this is histidine, and maybe this is then serine, and maybe this is then methionine, and yada, yada, yada. Or it's something else for a different protein. So the primary structure is how each amino acid lines up. I always like to think one by one by one. Kind of reminds me of little kids when they're like elementary school line and they have to form that nice straight line, right? Maybe Kyle's the line leader that week, and then Raina's the caboose or something, right? How they line up one by one by one. So then it starts to get a little complicated. We have secondary structure. And so this is formed by hydrogen bonds between amino acids. Oh, wow, I really put your amino acids there. So now we start to get like a little snake in our line, right? Basically, you can do hydrogen bonds, and this can result in two different types: a beta sheet, which is more like a crinkle, and an alpha helix. For the hydrogen bonding. So it can result in two types: beta, which is kind of like a crinkle, right? So here's a beta sheet, or an alpha helix, which is more like a coil. Okay. So we're still imagining our kids on the playground in their line, boom, 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 boom. But let's say, I'm making up names here, Susie, who's maybe third in row, all of a sudden wants to hold hands with me, and I'm maybe like 4 people behind her. So now our line is starting to get a little wavy, right? Because we still have that primary structure. We still have to line up in that certain line. So then we have tertiary structure. So we still have the primary, we still have the secondary, but now we're getting like all crazy. We're playing Twister. And so this happens because we have bonds between R groups. Right, so whatever that side chain is, maybe it's creating some kind of bond with another side group, or another side chain. And so basically you have 4 different kinds of bonds you can have between R groups. You have ionic bonds, hydrophobic interactions, you have hydrogen bonds again, but now this is specifically between the R groups. And you can also do disulfide bridges. Don't worry about really understanding what those are, just understand that tertiary structure is formed with bonds between R groups. So, back to our kids on a playground example, right? So they had to line up in a certain order, the one by one, and then, me and Susie wanted to hold hands, and so now we're starting to snake a little. Now all of a sudden, Tommy wants to high five me, and so now we're basically all playing Twister, right? We're starting to get this globular formation. We're starting to get all crazy and play Twister. So this is when the protein starts to become globular in 3D.
What's that last one characteristic?
Disulfide bridge. So here's an example of it here.
Thank you.
Say that again?
Oh.
Yeah, so here's a picture of those 4 bonds, or those 4, like the ways in which the R groups can then connect. You don't really need to know the exact names, maybe be able to recognize it in sentence. Disulfide bridge, oh, that's something to do with tertiary. So finally, we have quaternary structure, right, the 4th degree of Fulton. And basically, this is just 2 different subunits coming together. So, I know I'm beating a dead horse or something with my line example here, but right, we have our elementary school line. They were lined up one by one. They had a certain order. Then some of them wanted to hold hands, and we started to get some kinks. And then I wanted to high five all the way over here. So now we're getting all Twister. All right, so now say I'm part of Professor Daniel's class. Then all of a sudden, we wanna join our crazy twister line with Professor Cooper's class. That's quaternary structure, right? We had two different subunits that then came together.
What are the subunits?
The subunits are whatever went through primary, secondary, and tertiary together. Sometimes they're identical.
Are they already proteins or are they just amino acids that
They're already proteins.
are already?
Glucose is a carbohydrate though.
Oh,
So this is usually how proteins are depicted. Right, so we have some alpha helixes, this doesn't really have any beta sheets, but right, you have this big globular form, we got things binding in here, all crazy sorts of things happen, right? This has already gone through primary, secondary, tertiary, and quaternary structure. I found this picture in your textbook, which I thought was really fun. I never had thought about it this way, but you know, like an old office phone, right? So primary structure is just the core, how it lines up. And we let go, you know, they start to coil up. This could also potentially be beta sheets, but for this example, it's coiled. Tertiary structures when it starts to get globular and lay upon itself. And there's not an example here for quaternary, but quaternary would be like, I had this phone line and I pulled another one and I put them together. I thought that was a really unique example that I never thought of it like that.
I like how you said that's an old office phone.
Yeah, I knew as soon as I said
That's in my office right now.
I was like, people are gonna be.
It's got this brand new.
You got quarters.
It's voice over internet, it's voice over internet.
We got a boy.
It is.
See I mean, in our lab, we have like a lab phone, and it's just like a handset, like silent phone. All right, final couple slides here. Enzymes. Enzymes are a biomolecule, but even more specifically, it's a type of protein. So what the heck are enzymes, right? I said this is a special case of protein, right? Proteins are biomolecules. They typically end in ACE, right? So carbohydrates typically ended in OS. Here we have ACE. Enzymes speed up the reaction by lowering the activation energy. We already had this definition previously, but we called it a catalyst. Catalyst and enzymes, same thing. Right? So enzymes speed up reactions by lowering the activation energy. So if you remember this chart from earlier, right? We basically just lowered that hump, right? So it took less energy to do the reaction. So there's kind of two models that enzymes can follow. So we said enzymes are proteins, right? They end up taking this globular form, right? So this green here is your protein, your enzyme in that globular form, just simplified to an object, basically. Enzymes take on substrates. That's the reaction that they're trying to catalyze. So there's two methods, or kind of two rules of thinking for how these enzymes and substrates come together. So there's the lock and key method, which basically theorizes that the enzyme is an exact fit for the substrate. right? And so just to reiterate here, substrate is the reaction it's trying to catalyze, right? Active site is the location on the enzyme that binds substrate. Right? So basically, there's different enzymes that are unique to different reactions. And the whole idea with the lock and key method is that this enzyme will find what it needs to catalyze because it's an exact fit for the substrate.
What's that,
Substrate. It's the same word that's written right below it. Okay, or there, yeah. So our second model or second theory here is the induced fit model, which basically says that the enzyme will fit, but it's more like a hug. So the enzyme molds to the substrate. Okay. So the enzyme, the active site somewhat resembles substrate, but it doesn't exactly fit perfectly until the substrate is bound and the protein or the enzyme changes conformation, changes shape to fit that enzyme. So I kind of think of it as like a hug. It's like, come here. Oh, you're here. And so this model is more widely accepted by scientists. So there can be inhibitors, right? So we talked about there being a substrate and an active site that fits it. Well, sometimes things come along that somewhat mimic the substrate, but it's not the exact substrate for that enzyme. And these are inhibitors. So this is this is some other molecule basically. That mimics the substrate and binds to the enzyme. Okay. So it basically blocks it. So there's two different types of inhibitors, competitive and non-competitive. Competitive is going to bind directly to the active site. Where non-competitive are going to bind to some other location that's not the active site, but it causes a conformational change in the active site. So it's gonna be a really weird example, but this is what I feel like this is. So remember how we talked about like the enzymes are like, come here, give me a hug, you know, and then they're gonna bind. Well, let's say all of a sudden, like, so weird to say, but like somebody comes along and sticks something in your belly, you're like, ah, like, no, you're not ready for the hug anymore, right? So you've entered some area you're not supposed to enter, basically, and you've changed shape, and you can no longer bind to your substrate, right? You have something stuck in your asshole, you're not trying to get something out.
That's great.
Conformational change, right? So changing shape. So these inhibitors, sometimes we can pull them off the enzyme, right? That's reversible. It's probably bonded by non-covalent bonds, either competitively or incompetitively. But sometimes these inhibitors bind irreversibly and like, we're done. We can't bring the enzyme back, right? That's usually from a covalent bond. And in the case of cyanide poisoning, this is what happens. N minus, right? Cyanide particles bind to your enzymes, and you can't bind anything else. Your metabolic, your systems can't function properly.
Let's see, and maybe.
Yep. That's what cyanide looks like. It also was one of our polyatomic ions. I believe polyatomic ions. Molecule 2 or 3, I can't remember. All right.
What about the non-covalent?
So those are reversible. So sometimes those are things like ibuprofen and stuff. Those they go and block those pain receptors, right? And then you temporarily don't feel the pain or inflammation that you're feeling. But it's reversible, right? You then the medicine wears off and you can do all the other things again. You're trying to start to feel the pain. So the last slide here. There are factors that can either increase enzymatic activity or basically the rate of reaction. And so basically we can do that in 3 different ways. We can increase the speed of the reaction by increasing temperature. So if we increase temperature, we're going to increase the speed. pH. We can change the pH, and sometimes this will make the enzyme work better or worse. I don't have a general rule that if we increase pH, we increase rate, because the pH is unique to each reaction. But if we, let me write that actually, unique to each reaction. There's an optimal pH for each reaction. Another way that we can increase the rate of the reaction is to increase substrate concentration. So that's what this picture depicts here, is that enzymes are reusable. So we can have a substrate combined, and then it will release once it's done its reaction, right? Well, they're reusable, they're ready to go. If we add more substrate, we're going to end up pushing out more, right? So take home message here. We can increase, or we can change enzymatic activity by changing temperature, pH, and substrate concentration. All right. So we made it through, no videos. I'm happy too. All right, don't forget our last in-person lab tomorrow and that your initial discussion post is due tomorrow night at night.
You know, post those notes.
Yes, so, yes,
Any announcement?
yes, I'll make an announcement probably since I have all the other documents ready.
Thank you.
I gotta figure out how I can save them.
You can do it on notability, I don't know.
But like, if you don't have notability, like, you can make it a PDF, I can make it a PDF.
I can show you. If you don't.
Can you?
Maybe,
Teamwork.
they would know how to do anybody.
Cool, yeah, that was.
Look, see, you got lights on, how was that?
Stupid walking by.
Yeah.